5 similarities between mitochondria, chloroplasts, and bacteria cells, a double membrane structure. In bacteria, the plasma membrane serves as the site for crucial functions like energy production, while the additional outer membrane protects the cell. Similarly, the outer membrane of the mitochondria serves as a boundary, and the inner membrane is where ATP is produced. Chloroplast's outer membrane isolates the organelle, and the inner membrane regulates transport and houses enzymes. Genetic helps them produce proteins needed for energy production and photosynthesis. It also allows them to replicate independently, supporting their specific functions of the cell. Ribosomes produce proteins encoded by their own DNA, essential for energy processes, ATP production, and mitochondrial photosynthesis in chloroplasts. Binary fission ensures they replicate to meet the cell's energy or photosynthesis needs, maintain their numbers during cell growth or division, and replace damaged organelles, supporting overall cell function. Enzyme and metabolic processes. Enzymes drive metabolic processes by speeding up chemical reactions, enabling cells to produce energy, build molecules, and maintain essential functions efficiently.
Some benefits of the aerobic cell would be greater protection by living inside of a larger anaerobic host cell. The aerobic cell would gain protection from environmental hazards such as predation or harsh external conditions. Another benefit would be more efficient energy utilization. The host cell's metabolic byproducts could serve as substrates for the aerobic cell's oxidative metabolism.
There are two lipid bilayers around both mitochondria and chloroplasts that also help enhance the function of metabolism by creating distinct functional compartments. In mitochondria, by the intermembrane space, the region between the outer and inner membrane, where protons accumulate during the electron transport chain, creating the electrochemical gradient, the matrix where metabolic processes like the Krebs cycle take place, providing molecules for the electron transport chain. These compartments allow mitochondria to compartmentalize different biochemical processes, increasing the efficiency and control of energy production. In chloroplasts, the double membrane creates distant environments, separating the chloroplast's internal processes from the rest of the cell. This helps maintain the specific conditions needed for photosynthesis.
Theory 1 describes the creation of mitochondria, while Theory 2 describes the creation of chloroplast. In Theory 1, a larger prokaryotic cell absorbs smaller prokaryotes that produce ATP. In Theory 2, a eukaryotic cell that already has mitochondria absorbs a prokaryotic cell. This might have happened twice because chloroplasts and mitochondria are descendants of two different bacteria. Mitochondria coming from bacteria that uses aerobic respiration, and chloroplasts coming from bacteria that are photosynthetic.
The endosymbiotic theory has changed our view on how we see the complexity of life. It challenges the traditional views of evolution as a linear process, and it promotes the idea that life's complexities could arise from symbiotic relationships. This shifts our thinking about the nature of life, encouraging more collaborative and conjoined views rather than seeing organisms as isolated competitive entities.